Okay, so welcome back to Final DSE Biology Chapter 20. So in Chapter 20 and then also Chapter 21, we'll talk about more about molecular biology. So this is the beginning of what modern biologists are doing. So what exactly is molecular biology? So let's have a look. So in Chapter 20, we will first talk about photosynthesis. And in order to understand photosynthesis, firstly, we'd like to understand its role. So it is to provide the basic food source in the ecosystem. So based on the facts you learn in ecology, that's in chapter 19, and then you know that plants are the producer in the food chains and also the food webs. And also for photosynthesis, it will maintain the energy flow that is from the sun and then to all the organisms in the ecosystems. And for photosynthesis, it will also balance the oxygen and also carbon dioxide in the atmosphere. In order to detect photosynthesis, we detect the products produced. So for the two products produced by photosynthesis will be glucose and oxygen. For glucose, it is converted to starch. That's why we test the presence of starch. That is to use iodine. That's the iodine test, and the positive result is the color change from brown to blue-black. As for the oxygen, it is released as the byproduct. That is not the major product, but as a byproduct. That is something that you don't want, but actually they produce it. And the test is using a growing speed. The positive result will be it ignites. However, for oxygen, it is less likely to be detected because as oxygen is produced, usually water vapor is also produced, and the presence of water vapor will eliminate the light of the growing speed. So that's why it's not the best test in the world. In order to test for the presence of starch, we have to treat the leaves first before the experiment. That is what we call destarching. And the plants will be put in darkness for 24 hours. And it is actually something you learn in S2. And the process after before the iodine test is that first we will boil the leaves and then we treat the leaves with hot water bath with alcohol to remove the chlorophyll and also to dissolve the cell membrane, and then finally we will use hot water to wetten, and is to get it wet and also get it soft again in order to start the test. Otherwise, it will be too bitter. And for photosynthesis, actually there are some requirements like the carbon dioxide, light, and also chlorophyll. And in order to test this requirement, we use the setup as follows. For carbon dioxide, if you want to remove the carbon dioxide, we will use potassium hydroxide, that is KOH, in order to absorb the carbon dioxide. In order to test the presence of light, it's important for photosynthesis. We will use black paper or use some aluminum foil to block the sunlight. As for the chlorophyll, we will use the vegetated leaves. That is, it is not the not the whole leaf is green in color. And then we compare the starch produced in the green part and also the non-green part in order to test that chlorophyll is important for photosynthesis. And leaves are the major site of photosynthesis. That's why we like to understand more of the leaf. The leaves are designed to meet the following objectives: maximizing the absorption of light, facilitating transport of materials either into the leaves or out of the leaves, and also it would like to reduce the water loss. And it is a concept you learned in chapter 10 about transpiration. So actually there are a lot of overlapping between these two chapters. And here are some adaptations of the leaf. Remember, they are always complemented with one description and then one function. There's one feature, one function. So why leaves are very thin? Because it allows light and glass to reach the photosynthetic cells easily. So you can recall your memory in chapter 10. And similarly, this is also something you learn in chapter 10. For the upper epidermis, is usually with the palaeocytic mesophyll because they are closely packed in order to effectively absorb the sunlight. While in the lower part, that is the lower epidermis, it is commonly found with the sponge basophils in order to have some air space for gas exchange. And this is the classic terrestrial plant. So the upper side will be the upper epidermis, and it is usually covered with a cuticle to reduce the water loss. While for the lower side, usually a lot of stomata can be found because it will allow the gaseous exchange to happen while minimizing the gas and water loss. And in the middle, there will be xylem and phylum, and they will be used in transporting water and also the food, and it is something you learn in chapter 10 also. Back to the focus of chapter 20, we would like to focus on chloroplast. For the chloroplast, it is double membrane. It is a concept you learn in chapter 2. And for the chloroplast, now we would like to know it in more detail. So it contains the stroma, that is the fluid inside the chloroplast, and also there will be a network of membrane sacs that is called the photochemical, and also the granule. Granule is a makeup of a lot of photochemical. So for the structure of the chloroplast, usually the photochemical will be grouped together to form a granule, and then the granule will be commonly found in the chloroplast. It will produce a large surface area to pack more chlorophyll, and also the granule will be interconnected. Remember, these are all features, while the lower one will be the function. So when we talk about the adaptation, usually there will be one description or one features, and then there is a function. For photosynthesis, it can break down into two steps. The first step will be the photochemical reaction, also known as the light reaction, and the second step will be carbon fixation, also known as dark reactions. However, at the end of the day, usually we will not use light reaction and also dark reaction, and you have to understand why. For the photochemical reaction, if occur in the photochemical, that is on a stack like a sacs. It involve the excitement of the electrons in chlorophyll and also the splitting of water, that we call the photolysis of water, and as a result, oxygen gas is produced. As for the second step, that is the carbon fixation, it occurs in the stroma, that is the fluid part of the chloroplast, and it involves the synthesis of various molecules from carbon dioxide, and usually we will take the production of carbohydrates at the end of the photosynthesis. So let's understand the photochemical reaction in the greater details. So the absorption of light will result in the excitement of the electron, and then this excited electron will be passed down the electron transport chain, and through the process of photophosphorylation, the ATP will be formed, and ATP is the currency that we take as the energy. That is, if we're talking about the energy in cell, we will talk about ATP. And then the photolysis of water will also lead to the formation of NADPH and also the oxygen, because water contains H2O, and then the oxygen becomes the oxygen gas, and then the H will be combined with the NADP to be formed NADPH. And the second step of the photosynthesis will be carbon fixation, also known as the Calvin cycle. And for the carbon dioxide fixation, first step will be the carbon dioxide is bound to the 5C compound, and then this 6C compound will be split into two 3C compounds, because 1 plus 5 is 6, and then 2 times of 3 is also 6. And then this 3C compound will be reduced using the ATP and NADPH generated in the previous step, that is the photochemical reaction. And then this 3C compound will be turned to tau phosphate, and this tau phosphate will be used to form glucose. And this tau phosphate will also regenerate the carbon dioxide acceptor, that is the 5C compound shown in the initial steps. So let's use a graph to understand what is happening. So the carbon dioxide will bind with the 5C compound to form two 3C compounds. This is the first part, and then this 3C compound will be converted to tau phosphate in the expense of ATP and also NADPH. And then the tau phosphate will be converted to glucose. However, a lot of them will be converted back to 5C compound in order to accept the carbon dioxide again. And the conversion of tau phosphate to 5C is also expense of ATP. Here is the overall procedure of the photosynthesis. With the presence of light, water is split, and it will generate ATP and also NADPH and oxygen. And this ATP and NADPH will combine with carbon dioxide with 5C compound and generate glucose. And during this cycle, that is the Calvin cycle, ATP and also NADP will be regenerated in order to go back to here and combine with the water and then go to the carbon dioxide to start the whole thing once again. So in order to extend our understanding of the atoms in the products, we have to understand with these colored code. So for water, it is photolysis, that is the oxygen is split to form the oxygen gas. So this will be correlating to the production of the oxygen gas, while this H2 will be used to form the NADPH, and then NADPH will be used to form the glucose. So here will be corresponding to this one. And then as for the carbon dioxide, for the C, it's definitely go to the glucose. So this corresponding to the green side here. And then for the oxygen, it is also entering with the glucose. So traditionally, we would like to compare and contrast photochemical reaction and also Calvin cycle based on the time, place, energy change, inputs, and also output. And it is already found in a lot of essay type question or in the past paper. I strongly recommend students to have a look in those questions to be familiar with this topic. And let's talk about the fate of the products. So tau phosphate is the primary product, and then tau phosphate will be used to synthesize fructose and also glucose. And with fructose and glucose combined together, it will form the fructose and glucose combined together will form the sucrose. That is a common form of carbohydrates used to transport in plants. And then a lot of glucose combined together will form the starch and also cellulose. For starch, it is commonly used as a storage product, while for the cellulose, it is used as the structural materials to form the cell wall. But how about lipid and also proteins? Lipid and proteins are produced through the intermediates of glucose breakdown, and then they will generate the glycerol and also fatty acid. For glycerol and fatty acid, they will be converted into lipid, while for amino acid, it will combine with inorganic ions to form protein. So what are the inorganic ions mentioned here? I recommend students to have a thought about this. So finally, we would like to talk about the factors affecting the rate of photosynthesis. There will be generally two factors, that is the light intensity and also carbon dioxide concentration. As for the effect of light intensity, for low and medium level of light, there is an increasing trend. However, up to a moment, there is a saturation point, and then the effect is levels off. And then in a very high light intensity, the photosynthetic rate will be reduced. So what is the limiting factor that results in the saturation point? Usually it will be the carbon dioxide concentration, temperature, or other factors. Because in this setup, we are only monitoring or we are just manipulating the light intensity. That's why the carbon dioxide should be remained unchanged. So how about carbon dioxide concentration? So here are two graphs. The purple one corresponds to a higher carbon dioxide concentration, while the orange one corresponds to a lower carbon dioxide concentration. You may notice that both of them will be leveled off to a certain level. However, you may notice for the low carbon dioxide concentration, the maximum photosynthesis rate is much lower than the higher one. And also, the saturation point for the lower one will be reached much sooner than the higher one. So, you have to understand how we may find similar and also difference in these kind of graphs. So in order to regulate the rate of photosynthesis, we humans invent greenhouse, because in greenhouse we can monitor temperature, humidity, carbon dioxide, and also soil condition. That's why we can also reduce the damage by pests, and we can have crops throughout the whole year. So this is a very classic pictures that I would like to share with you. So in Form 2, you learn about photosynthesis, and now you also learn about photosynthesis, and I bet that your face should be looking like this one. However, in the university, if you have a chance to come across with photosynthesis once again, you may notice that currently you are in such an easy mode of photosynthesis, and I'd like you to have a notice of this. So you should be aware that we are not demanding too much from you guys. So here is an extract of the HKEA document. We'd like to understand photosynthesis as the site, that is the leaf, and also the requirement of photosynthesis, the light, carbon dioxide, water, and also chlorophyll. And as you are senior students, we would like to break down the photosynthesis into two parts, that is photochemical reaction and also carbon fixation, which is also known as the carbon cycle. And finally, we would like to know some factors affecting the photosynthesis rate, that is the light intensity and also carbon dioxide concentration. So here is what chapter 20 is about. So I hope you will like this video. See you guys next time. Bye bye.